Hi, everyone. Okay, this is part two of this week's lecture notes. Uh, in part one, earlier this week, I talked a bit about bacterial division. They do divide by binary fission. And I also talked about bacterial growth, specifically growth within a population of bacteria, and the phases of growth in a population under closed conditions, under limited nutrition. Now I'm going to continue about talking about environmental factors that are also important to bacteria for growth. The first one there at the top, biomolecules or food, we all know is very important. And why? Because the bacteria need the energy and carbon from the food to help it for reproduction. Now, there are other chemicals and physical factors in their environment that also support bacterial growth. And the four are oxygen, temperature, water, and pH. Oxygen is needed for the aerobes or other alternative electron accepting gases for the anaerobes. Temperature, we're talking about a temperature that will permit optimal growth. Water, we know bacteria need water, but also in addition to water, which goes hand in hand with the water, is the appropriate osmotic environment. And finally, the pH. And for pH, it isn't always right to say neutral, because bacteria we find are everywhere, and there are also bacteria that live in different pHs or need different pHs in order to grow. So anyhow, talking about number one, oxygen, what we know is in the aerobic life, specifically aerobic respiration, oxygen is needed as the final electron acceptor in the electron transport system. Now, a certain percentage is oxygen, maybe about 2%, becomes reactive oxygen species or becomes molecules that have unpaired electrons. So if you look at the diagrams there on the right, we see oxygen nice and in balance. All the electrons are there. It's all in blue dots. But if you look at peroxide, superoxide, anion, and hydroxyl radicals, those red dots are representing unpaired electrons. What does that mean? It means that that molecule is very unstable. It wants another electron. So it takes it. It'll take it from wherever it can steal it from. And usually that will be from molecules that make up the bacterial cell, from DNA, from whatever's available. And that's why these are called free radicals. They're free molecules, and they will pretty much rob these extra electrons in order to become completely balanced. And again, in the meantime, they are, over the long run, they damage cell structures, they damage tissue in organisms, higher organisms. They also will cause damage to DNA and proteins. So, how does a system that undergoes aerobic respiration cope with this? It's able to cope with this because it has a defense system, mechanisms, vitamins, enzymes, and other proteins that can counteract this free radical that is produced. So if you think of in our lab with the catalase test, that is one example. That enzyme will take peroxide again and convert it from a free radical into water and oxygen, a balanced molecule of oxygen. if you look at these three that I've talked about here, the free radicals, they are grouped into a group called reactive oxygen species. And all that means is these are oxygen molecules that are very reactive, very unstable, and in the long run, not very good for the cell or the organism. Now, anaerobic bacteria, which undergo anaerobic respiration, there's a lower price. They don't form these free radicals because it's an inorganic molecule that will accept that final electron. So there is a lower price, but then again, they do pay a bigger price if oxygen does become available in their environment. And I'll show you that at the next slide. All right, so this was a picture that was in your lab, I think, a week ago or so. And there are 5 groups or 5 categories, depending upon the oxygen need of bacteria or prokaryotes. And we've got your obligate aerobes, obligate anaerobes, facultative anaerobes, and aerotolerant anaerobes, and finally microaerophiles. So the ones that can survive or do fine in the absence of oxygen are going to be your obligate strict aerobe. And again, this type of bacteria really does not want any part of oxygen. In fact, if they are exposed to oxygen, will die. The reason for that is even though they undergo anaerobic respiration, they do not have any of that defense mechanism to basically work on the free radicals that are produced if oxygen is in the air or the environment. Facultative anaerobes and aerotolerant anaerobes do okay with oxygen because they have some of these enzymes that we're talking about that fight that fight the free radicals. And if the oxygen is present, we've got the obligate aerobe, facultative anaerobe, aerotolerant anaerobes that can do fine in the presence of oxygen. And for the same reasons that they do have some defense system in place to counteract free radicals. Okay. All right, temperature is another environmental factor, and with temperatures, we've got bacteria that like different ranges of temperature. There are some that will grow in very, very cold environments, and some in very hot environments above boiling. So there are 4 groups here, and the psychrophiles, the mesophiles, thermophiles, and hyperthermophiles. The psychrophiles, the end of the word phile just means loving. So they love or grow very well in these temperature ranges. The psychrophile loves very cold temperatures. It may grow in temperatures as low as 0 degrees Celsius. But optimum is about 14 degrees Celsius, so they grow best at about 14 degrees Celsius. Your mesophile, meso meaning middle, they love to grow between the range of 20 to 40 degrees Celsius. The optimal is 37. So this is, by the way, where many pathogens grow within this range, because of our body temp being 37, they will grow best at 37. Now we've got the thermophiles, the next group. They grow at a little higher temperatures, 55 degrees Celsius and higher. So they grow between about 40 and 70 degrees Celsius. They like it a little warm. And then finally, growth as temperatures as high as 121. That will be your hyperthermophiles. So they grow between about 65 to 121. And in this group, the hyperthermophiles, this is archaea only. Okay, archaea only. And I have a little fact here. If I would take that 121 degrees Celsius and convert it to Fahrenheit, that equals 250 degrees Fahrenheit. So that is at quite a high temperature. Now, do we see these temperatures? Sure we do. We see them in the hot springs of Yellowstone National Park. This one species of bacteria, Thermus aquaticus, was discovered there by Thomas Brock in only the 1960s. And then you see also thermophilic and hyperthermophilic lithotrophic archaea. So these are rock eaters. And these are discovered in the 1970s, and they were discovered in hypothermal vents, which are found at the bottom of the ocean. Basically, they're like volcanoes undersea. And what happens with these thermal vents is that the cold water will enter them from the ocean bottom, and it is heated up immensely by the magma, and then it is released out of those vents. But they have found bacteria living there, and also eating the sulfur that's down there in those vents. So we're finding bacteria everywhere. Okay, another environmental factor is water. And with the water, I'm talking about also osmotic pressure. So it's going to be water. In this case, the solute will be salt. It's important to keep a consistent salt concentration for the bacterium. And it depends upon the bacterium, because some like higher salt content than others. If you do not keep a consistent salt concentration, it will basically damage the cell by either making the cell lose water or having too much water go into the cell. The cell wall will protect the bacterium to a certain extent, but not in extreme conditions. So what we have here, we've got three groups of bacteria. We've got the non-halophiles. The non-halophiles, which they do not, they usually can live within 0.2% to 5% sodium chloride environment. We've got the moderate halophiles, and these are basically your marine living organisms. They can tolerate 5 to 20% sodium chloride within a water environment. And then finally, the extreme 20 to 30% halophile. And these, again, these extreme halophiles are your archaea. So again, halo means salt, phile loving. So if you see the word halo, we're thinking salt concentration. Okay. And anyhow, there are some examples of that. On the right-hand side there, we have halophilic and thermophilic and radiation-resistant archaea living here. So we have three different kinds of archaea, or archaea that can be halophilic and thermophilic. And these are in the salt flats in Nevada. Okay, the colors are the result of the pigment of the bacteria. Now, how do these how do these bacteria, how are they able to cope with high salt concentration especially? And it's because they've got special pumps or mechanisms that can keep the internal concentration within the cell at a normal range. So these guys come with stuff that enables them to live in these higher salt environments. pH is another environmental factor. And again, we've got three groups here. We've got your acidophiles, which live in acidic environments. Those acidic environments are usually the result of strong acids that they produce, that the bacteria and archaea produce by oxidizing metals. Then we have neutrophiles, which are bacteria and archaea. And again, this includes a majority of human pathogens because these pathogens can grow within our body pH. And then finally we have alkalophiles, which are alkaline bacteria. They grow in pHs above 7, a little basic. And these are found in the soda lakes that I just talked about. So anyhow, here's an example. Here's an example of acidic drainage from a coal mine where you see acidic bacteria and archaea. And again, this is a spirulina aquatic culture. All right, spirulina is a photosynthetic bacterium that grows in alkaline conditions. And this is actually, spirulina has actually been found to be very nutritious. It's been packed with protein, B1, B2, B3 vitamins, and copper and iron. It was used in cattle feed and fish food, but now they have discovered after the NASA mission, that this is a very good nutrient they had fed to the astronauts, and it now can be found in a lot of health food stores. But anyhow, that is us making a market out of some knowledge we have found out about certain bacteria. Anyhow, those are the four environmental conditions that aid in the growth of a bacterial population. And again, this you probably would be asked on an exam as far as the names to each of those conditions, the categories of each of the bacteria to those conditions, those environmental conditions. So anyhow, I'm going to end for now. I hope you all have a good weekend, and I will be talking and posting more for next week in a little bit. Okay? Have a good night.